Okay, today I'm going to go over bacterial virulence factors with a focus on E. coli virulence factors, hopefully in 10 minutes. So, what is a virulence factor? According to the highly regarded scientific journal Nature, virulence factor is the intrinsic character of infectious bacteria that facilitates their ability to cause disease. Pretty vague, but hopefully we'll be able to nail that down in this presentation. Virulence factors can be broken down into two broad categories. In general, virulence factors can be factors that are released by the bacterium that actually directly damage the host, or they can be protective factors that allow the bacterium to avoid host destruction and continue causing disease. So you can see my macrophage on the right side that's trying to engulf or phagocytose bacterium, but it's being protected by a virulence factor. So in order to keep this presentation relatively short, I'm going to focus on E. coli. E. coli is a gram-negative bacterium, which means that the outer membrane here protects the layer of peptidoglycan that is used in gram staining. The stain that's used is crystal violet, which will bind to peptidoglycan in gram-positive bacteria, but because of this outer membrane here, none of the crystal violet will be able to get through. So the peptidoglycan layer, outer layer of plasma membrane, and an inner layer of plasma membrane. Also important is what's called endotoxin or lipopolysaccharide, which is embedded in the outer layer. And endotoxin is made up of an O antigen, which is the outer part, followed by a common core polysaccharide, and this is widely conserved across many gram-negative bacteria. And then the lipid A, which makes up the base of the LPS, of the lipopolysaccharide. So this whole thing together is called endotoxin or lipopolysaccharide. In addition, there's another antigen for antibodies and the host immune system to attack, and this is called H antigen right here, which is a common motif on many flagella and other external structures of the bacteria. It's also important to note that E. coli are facultative anaerobes, so in both O2 rich and O2 poor environments, they are happy. Factors that enable the E. coli to avoid detection include the K capsule. In terms of virulence factors that help the bacterium to continue to survive and therefore continue to cause disease in the host, there's the capsule, which is a slimy layer of carbohydrates that surround the bacterium, and this prevents macrophages from engulfing the E. coli. It's also important to note that because the K capsule and the H antigen on the flagellum, so the flagella here, and the capsule itself are under control of of various genes within the bacterium, there is something called antigenic phase variation, in which the bacterium will change the different antigens to avoid detection by the immune host. So it has different targets covering it, and therefore it'll be in disguise and be more difficult to find. In terms of the ways in which E. coli can directly cause disease to the host, we have the LPS that we mentioned earlier, which is made up of lipid A, which is embedded in the plasma membrane, the core polypeptide, and a O antigen, which is what various antibodies recognize on the cell surface. During growth or lysis of the bacterium, the LPS is released and binds to CD14 on macrophages, which causes a very strong reaction, a very strong inflammatory reaction that can actually be negative and detrimental to the host. There's also something called the type 3 secretion system, which is a way for the E. coli to inject its own virulent proteins directly into host cells. So in this diagram we have the E. coli here, and these are host cells right here, and this represents the type 3 secretion system, which actually directly injects its virulent proteins into the host cells where it can do damage. Depending on the proteins that are injected by the type 3 secretion system, there are various pathologies that result, and each of the different pathologies classify the E. coli into different categories. So we have enterotoxigenic E. coli, which invades the small intestines and actually causes a diarrhea called traveler's diarrhea, which is caused by a heat-stable as well as a heat-labile enterotoxin that is secreted into the enterocytes, the epithelial cells of the small intestines. There is enteropathogenic E. coli, EPEC, which has a similar pathology. It's plasmid-mediated, meaning that it's the category of E. coli is created by a specific plasmid that's transferred into the E. coli. And it's categorized by damage to the microvilli of the small intestines, which leads to malabsorption and diarrhea. There's also enteroaggregative E. coli, which is again plasmid mediated, so it receives a plasmid to become this form of E. coli, and what happens is that it actually leads to hemorrhage as well as decreased fluid absorption in the small intestines. And these are particularly, this form of E. coli leads to sepsis often. Enterohemorrhagic E. coli, injects what's called Shiga toxin through the type 3 system, and this leads to lesions in the microvilli and decreased absorption as well. And this form of E. coli also is highly involved in sepsis. And finally, entero-invasive E. coli leads to general epithelial destruction of the lining of the colon. Uropathogenic E. coli are defined by being opportunistic infections of one of these other forms of E. coli, and they're generally another form that receives a plasmid that allows it to bind to the epithelium of the urogenital tract, and the specific adhesin or specific antigen that allows for the binding to the urogenital tract is called P. pili. So any of the other forms of E. coli that actually receives a plasmid with P. pili can cause a urogenital infection if it actually gets into the tract. So that's all for my presentation. I hope you learned something about bacterial virulence factors, especially in E. coli. Thank you.